Santiago. I'm a final year PhD student at Warwick University. I'm going to talk about how swarming bacteria can transit into biofilms in presence of antibiotics. Swarming bacteria are bacteria that travel in groups instead of as individuals. As you can see here, they travel in small rafts, and the interesting thing about these bacteria is that they can resist better antibiotics than when they are swimming. From a physics point of view, these bacteria can be modeled as a liquid-like phase of active particles, so they can be represented as a phase diagram of cell speed and cell density. According to the theory of active particles, any active system that has enough high speed and enough high cell density undergoes what is called motility-induced phase separation, which happens in local regions where cells slow down, and due to this they phase separate to something that is liquid-like to something that is more like solid-like. These made us think that actually these swarms can transit into something that is more static, a form of bacterial communities that are more static called biofilms. And this is interesting because biofilms are actually even more resistant to antibiotics than swarms. To test this hypothesis, what we did is we placed an antibiotic disc in one side of a plate. We let it there for 24 hours so it could create a gradient of antibiotics, and then we inoculated our bacteria Bacillus subtilus in the center of the plate. This swarming bacteria spreads until it encounters the presence of kanamycin, and to our surprise, what happened 40 hours later is that at the very front we saw wrinkle formation. And wrinkles are the morphological feature typical of biofilm formation. So we can actually see that these swarms form biofilms as it was predicted with this motility-induced phase separation theory. And this just happens in presence of antibiotics, since here in the bulk we don't see biofilm formation. To test whether these swarming bacteria were actually following motility-induced phase separation, we tracked the system in this cell phase space. We measure the cell speed and the cell density, and what we observe is that for certain combinations of this cell speed and cell density, bacteria started to form jams. These local jams can be observed in this video, where the velocity of the bacteria were measured, and you see a drop at the same time that bacteria actually form this gray region called jam in the screen. Eventually, these jams give place to what we call the islands, and these islands are localized regions where bacteria actually go to second layers. You can see here in this video where I show a microscopy view of the whole plate, and you can see how this island formation happens at the very front near the antibiotic disc, and it doesn't happen in absence of the antibiotic. Actually, these islands then develop multiple layers, so we can say that actually this kanamycin disc or this antibiotic disc triggers this heterogeneous cell density, and this was actually related to the formation of the biofilm. So after this heterogeneous cell density, we can observe the formation of wrinkles here at the front, the same place where the multilayer is formed. This transition is actually quite dangerous, since something that is 10 times more resistant to antibiotics can transit to something that is even more resilient to antibiotics. We know that the key step for this transition from swarm to biofilm is in this heterogeneous cell density, so we thought, what if we can actually target this step? To do so, what we did is we split the total amount of kanamycin in 2 time points. The first time point will trigger this heterogeneous cell density as we observed before, and the second time point we will add a second dose of kanamycin. And this is what happened. When all the kanamycin was added at the first time point, as I showed before, wrinkles were formed. Then when a second dose of kanamycin lower than the first one was added in the second time point, wrinkles were still formed. When the doses in the first time point and second time point were equal, the wrinkles started to fade away. And then finally, when the second time point concentration was higher than the first one, the wrinkles almost disappear. So we can conclude that actually, when we split the kanamycin in 2 doses and the second one is higher than the first one, we can inhibit wrinkle formation and biofilm formation from swarms. Thank you very much for your attention. I hope you enjoyed the presentation.